Buenos días. Continuando con las presentaciones, ahora corresponde la presentación del profesor Jonathan Gibbins de la Universidad de Reading, de Inglaterra. El profesor John, su principal línea de investigación son estudios de función plaquetaria, muy importante en la hematología, estudios plaquetarios y cómo participan las plaquetas en la trombosis y su prevención. Además, bueno, hemos tenido la oportunidad también de estar en el centro del profesor Gibbins en Inglaterra en un par de oportunidades, realizando algunos experimentos, y durante esta semana hemos tenido varias reuniones de trabajo con el profesor para discusión de proyectos de investigación. Así que muchas gracias, John.
Thank you very much, Eduardo. I'm going to take this off. So thank you very much for the invitation. It's a real pleasure to be here and get to experience life in Tauca and find out about the university. As Eduardo says, we've been working together for a while, and so it's really good that we get to meet, and I've had a fantastic week meeting all of the group and talking science and thinking about things that we could do in the future. I need to apologize. You probably gather that my Spanish isn't very good. And so I apologize for the fact that you're all having to listen to me via a translation. But I do want to thank the colleagues in the room at the back who are doing probably the hardest job of all, which was translating Spanish into English and now English into Spanish. So the other funny thing is that I've now asked them to thank themselves. So there we go. So what I wanted to do today was introduce to you some work that we do in my lab. And I realized that we have people here from different backgrounds and different experience of the science. So I'm going to introduce some general ideas, but I think first of all, I need to give you a little bit of context as to what we've been studying. And the title of my talk is Mind the Gap, Surprising Ways That Blood Cells Talk to Each Other. And the work in my lab is concerned with understanding how cells work. I'm a cell biologist. So we do work that really tries to understand how things work, how cells work, and in this context, how they control blood clotting. But we also do work to understand how this contributes to disease, and also work to look at patients and to try and understand how we could use our discoveries to better treat patients and make them reduce the risk of serious disease. But first of all, where am I from? So if any of you have been to the UK, this is a map of the UK. I don't know whether you can see my... I've lost my cursor. Oh, there we go. There we go. So Reading is just there. So we're right at the bottom of the UK, and there's London right next to it there. And some of you may, if you've travelled through Europe, may have found yourself in Heathrow Airport. That's just next to Reading. So if you do ever find yourself nearby, do let me know. It's a long way away. It was a long trip, nearly 12,000 kilometres, but very straightforward. The University of Reading is quite similar to the University of Tauca. It's very green, lots of open spaces. And we're really lucky. We've just got a brand new building that we've been moved into, and we have new laboratories. We have a zoology museum, so we have all sorts of biology going on, and some really nice laboratories. And we also have some important guests occasionally come to see us, and there's Eduardo in the lab wearing his... he does wear a lab coat when he's in the lab, which is nice to see. So the context of what I want to talk to you about is the process of hemostasis. This is blood clotting, effectively. And we're interested in this because, as you'll see in a moment, it's associated with disease. But first of all, I wanted to show you what happens normally. So this is a platelet here. And it's recognized damage to a blood vessel wall. So there's damage to this blood vessel wall. And it sees the extracellular matrix or proteins in the blood vessel wall and sticks to them. It does this indirectly via other proteins that are in the blood. And this enables the platelets to see tissue damage. So they recognize when the inside of a blood vessel is damaged, and they change their behavior very quickly. So platelets are the smallest blood cells in your blood, and they're able to recognize injury. And they go through this process of activation. And when they become activated, they become sticky and they stick to each other. And you can see they're starting to stick to each other here. But importantly, they send out signals to other platelets to tell them that there's damage that has occurred. And these signals are able to bind to particular proteins or receptors on the surface of other platelets. And this tells them that there's damage going on. Ultimately, what this leads to is the formation of what we call a thrombus. It's the place where lots of platelets have stuck together and they literally plug the hole. So they stop the blood from leaking from the damaged blood vessel. So clearly this is really, really important. If our blood didn't clot properly, we would leak when we cut ourselves, and quite small injuries would be catastrophic. They would cause serious problems. But there's another side to this, unfortunately, a rather sinister side to platelet function. Some of you may recognize what this is. This is the heart, and we're going inside a coronary artery. So this is a blood vessel that supplies the heart with oxygenated blood. So this is the blood vessels. These are the blood vessels that enable the heart muscle to work properly. The problem is they're very small, and they tend to develop atherosclerotic lesions. And these are the end point, if you like, of the story that Professor Salazar has just introduced us to, looking at statins that reduce cholesterol levels, because high levels of cholesterol lead to the formation of these inflammatory sites inside blood vessels. The problem with them is that they look like damage to to platelets. And these little purple things here are platelets that have adhered themselves, they've stuck to the inside of these lesions because they look like injury. So the platelets are doing what they should do, but they're misrecognizing what they're seeing. And what tends to happen is they respond in exactly the same way as they would at a site of injury, but rather than plugging a hole, they clog up the blood vessel. They stop the flow of blood within the blood vessel. And this is the principal cause of heart attacks and strokes, which are the major killers in our societies. So the motivation for my whole field of research is understanding how this works, to see if we can develop better and safer drugs that are able to protect or reduce or treat individuals who have heart attacks or strokes. So we're interested in platelets and how they see what is going on around them, how they recognize tissue injury, and how maybe they ignore things that look normal because we don't want them to activate when they shouldn't, for the reasons I've just shown you. Now, we know a lot about how cells communicate, and this is an area of science known as intracellular signaling. And what we know happens in most cellular systems is that one cell might release a signal that binds to a receptor on another cell, and this makes that cell change its behavior in some way. And so the signals come from one cell to another. It takes a little bit of time. In some cases, it may be a surface. It might be the extracellular matrix that is the trigger. But it does this by binding to receptors on the cell surface. If a cell doesn't have the receptor for that particular molecular message, it can't respond. And we tend to draw, as scientists, we tend to draw out these long pathways of events that then happen to explain how the cell then changes its behavior, hopefully in an appropriate way. So this, believe it or not, is a simplified summary of a number of receptors and key molecules inside platelets that control their function. So almost everything we know about how platelets recognize tissue injury and respond involves intracellular signaling. So the pictures I've just shown you in the movie showed platelets releasing factors, releasing chemicals that activate other platelets. That's a process of intracellular signaling. And the reason this is really important is if we look at most of the drugs that are used that are relatively successful, they're not perfect, but they're relatively successful in treating or preventing heart attacks and strokes, they target the receptors for the things that platelets release to communicate with each other. So this is really important, but we realized a few years ago that maybe there are other processes going on, other ways that cells, platelets, might talk to each other. So on the left-hand side here is actually a technique that I'm going to show you a few times in my slides. This is known as intravital microscopy. And what we're looking at here is inside a very small artery in a living mouse. And we've injected this mouse with an antibody with a fluorescent dye attached to it, so we can see where the platelets are. Then what we've done is to trigger thrombosis. So this is bad clotting, if you like. And we've triggered this by firing a laser down the microscope at the inside of the blood vessel. And this triggers the thrombosis to occur. I'm showing you this because I'm going to come back to this a little bit in the future, in my subsequent slides. But what we realized, and others started to realize, was that platelets carry on talking to each other when they formed this thrombus. And that's important for the blood clot to work properly. And we've spent a lot of time looking at various cell surface receptors and conventional ways of looking at how cells communicate. But what we realized was that maybe the cells talk directly to each other. They don't send signals to the outside of the cell, but they actually talk directly because they're connected to each other when a blood clot forms. And so this is from a review from Skip Brass's lab, but it shows platelets that have stuck to each other in a blood clot, and they're touching via all of these different receptors that enable the cells to carry on talking to each other. But if you look in a textbook at the various ways that cells touch each other and can communicate with each other, there are a whole range of different processes and structures that form. But we were particularly drawn to these structures here, channel-forming junctions or gap junctions. These are channels, as we'll see in a moment, that connect the inside of one cell with the next cell. And these are formed through proteins called connexins. Connexins are transmembrane proteins, so they go across the membrane of a cell, but they form these really elaborate six-membered rings, which forms a pore in the cell surface. But where you have cells next to each other that both have connexins on their cell surface, they join together and they form a pore-like process or a gap junction that joins the inside of one cell with the inside of the cell next to it. And this enables small molecules, they are very small molecules, but up to about 1,000 Daltons in size, to pass from one cell to the next cell. And this is a different way of communicating. This is known, it's been known for some time, it's important in lots of tissues. Probably one of the best examples is cardiac muscle, so heart muscle. And one of the reasons why the heart contracts in a coordinated manner is because the cells directly talk to each other. And the signals that make the heart muscles contract are able to pass between individual cells through the gap junctions that are between the cells. So, what gap junctions do is they enable messages, molecules, small molecules, to pass from one cell to the cell next to it. But they can also work as channels on the cell surface and enable things to go in and out of cells. As you will see, we don't know very much about how these particular channels are controlled, but that makes them interesting. So we had a few clues that connexins might be important in platelet function. What you're looking at here is platelets down a microscope, and the platelets have been labelled with, they've been stained with a dye, a fluorescent dye that is sensitive to calcium. And the levels of calcium inside cells is fundamentally important to regulate a whole range of different processes. And so hopefully you can see on the left-hand side the platelets flashing on and off. And they flash when the calcium levels go high. And when the calcium levels go low, they stop flashing. But if you look really carefully, you might be able to see platelets that are sitting next to each other, flashing on and off at roughly the same time, which suggests that they're sharing signals between them. Now, we don't know whether that's involving gap junctions, but it was one of our initial thoughts, and it made us look in a little bit more detail. So we looked at platelets, and actually, initially, we looked at the cells that produce platelets. Platelets are actually difficult cells to work with because they don't have a nucleus. So a lot of the genomic techniques that you might use with lots of other cell types are quite difficult if you're working with platelets. But we can look at the cells that make platelets from the bone marrow, and they're called megakaryocytes. And this is RNA profiling. So this is looking at all of the genes that are being expressed in these particular cells at a given time point. And we can see here that there are 3 connexins that are quite noticeably expressed. And so we started looking to see if they're involved in platelet function. And importantly, we were able to show, this is an analysis where we're just staining the proteins from extracts from platelets, and we were able to show that platelets have each of these individual proteins. Now, there's quite a lot known about connexins. They've been studied for a long time, but not in blood cells. And several of them are expressed, or they're found in lots of different tissues. But I'm going to focus more on connexin 62 today, because almost nothing was known about that until about 2 years ago when we published the first paper showing what this protein is for. Just to make it slow, the numbers refer to the size of the protein. Okay, so connexin 62 is bigger than connexin 37, but they all work in a similar way. To be slightly confusing, in the mouse, connexin 62 is known as connexin 57. It's just a little bit smaller, but it's the same protein. So we wanted to know whether we could see connexins in platelets in the right sort of place you'd expect them to function. And so what we're looking at here at the top is platelets before they've seen anything that activates them. And on the left-hand side in green is a platelet receptor. So it just tells us we're looking at platelets. And we can see that they're nicely stained on this confocal microscopy image on the cell surface. If we stain for connexin 62, we can detect it quite clearly. It's mostly inside the cell. But when we activate platelets with things that they would see at sites of tissue injury, the protein moves to the cell surface. And this means that it's located in the place where it could then form gap junctions if platelets were able to touch each other. So if we allow platelets to clot and then fix them and then make very thin sections of them and do electron microscopy, we're able to look for features and see whether they look a little bit like gap junctions. And one of the features we tend to see, I'm not sure how clear this will be to you, but points where the membranes between platelets touch, in some places they come very close together. And this is a feature that you often see where gap junctions are formed. So this, again, gave us a few clues that gap junctions might be there and might be important. But in order to actually study these, we need to look at what they might do. And in order to understand what they might do, it's important for us to be able to inhibit them, i.e. prevent them from working, or as you'll see in a moment, we can also remove them from cells as well. So there are a number of ways that this is done. There are chemical inhibitors, and these are a couple of them here, that are used widely, but they do other things to cells. They're quite toxic. And then there are what we call mimetic peptides, and these are peptides that are designed against the sequence of the connexins that are thought to interfere with how they interact with each other and prevent the gap junctions from working properly. And they're called gap peptides, and the numbers after them just refer to where they are on the sequence of the protein. And the ones that we focused on are all called gap 27. And so these are selective inhibitors. They enable you to block the function of specific gap junctions on cells. And then the other thing that we have at our disposal is transgenic mice. So we're able to genetically modify mice so that they lack particular proteins. And so a lot of the work we've done is focused on mice that lack connexin 37, 40, or 62. And we're currently working on one that just lacks the protein in platelets, and that's ongoing. So just to look very quickly at the peptides that we've produced, because as I mentioned earlier, connexin 62, no one knew what it did. It was a brand new, newly recognized connexin. So what we've done is to devise a sequence for a peptide which we are able to then use to inhibit connexin function. And we've also been able to model the 3D structure of this particular connexin, which then enables us to put this all together and predict what the peptide might do to a gap junction. And this is done through docking experiments. So this shows you the ring structure of 6 connexins that are linked together. And there's a hole in the middle. And that hole is literally where the small molecules go in and out and pass from one cell to the next. And we're able to see where these peptides might bind. And what we think happens is that they cause a conformational change in the protein, which causes a loop to flip out and to block to basically block the hole and prevent molecules from moving from one cell to another. So what might these do? This is a really simple experiment, and I wanted to include this on here because I know that Eduardo's lab uses a similar technique for a completely different reason, so I think that's quite interesting to highlight. What we did was to load platelets with a fluorescent dye. So there are various ways of doing this, but we can get dyes to go into platelets and then get trapped there, so they can't escape. Then what we did was to stimulate them and ask whether the levels of fluorescence inside those cells changes. We do this with a technique known as flow cytometry. And what we were able to see is that when you activate platelets, you have a reduction in the level of the dye inside them, which suggests that pores are opening and the dye is leaking out. But if we do this in the presence of our peptide that inhibits the connexin, it stops them leaking out. So this suggests that the connexins on the cell surface are able to open and close and let molecules out. I mentioned that we've made a transgenic mouse. I'm not going to focus on this at all, just to say everything else in these mice is completely normal. They just lack this particular protein, and there's no change in any of the other proteins of interest on platelets. So what happens if we stimulate platelets, try to make them activate in the presence of inhibitors of this particular connexin? Well, this is known as platelet aggregation, and what we're doing here is measuring the optical density of a suspension of platelets in plasma. So the platelets, if we isolate them from blood, look sort of cloudy. If you look through them, they look cloudy, but when they activate, they stick to each other. They aggregate just like they would inside a blood vessel that was damaged. And when they do that, the amount of light that passes through the suspension of the cells increases. And that's what we see here as the trace goes down. But what hopefully you can see is with increasing concentrations of our peptide inhibitor, we're able to prevent platelet aggregation. So this suggests that connexins are important for regulating platelet aggregation. And you can see that graphically here. The interesting thing is that as platelets start to aggregate to each other, what we could be looking at is the function of gap junctions, but we could also be looking at the function of the individual channels on the surface of platelets, just letting molecules in or out of the platelets. So in order to look at this in a bit more detail, we can measure platelet activation, but using flow cytometry. And this enables us to just focus on individual cells, cells that aren't stuck to other cells. And what this enables us to see is that platelet activation is inhibited quite dramatically, even on individual platelets. So this suggests that the proteins act as pores or channels on the platelet surface under normal circumstances. On the right-hand side here, you can see a comparison between a normal mouse platelets and platelets that lack connexin 57. So you can see when we remove the protein from platelets, it also has a dramatic effect on their function. So this gave us a lot of evidence that this might be important. But we wanted to know how this might work. Can we find evidence that platelets do talk directly to each other? So in order to do this, we've used a technique known as FRAP, or fluorescence recovery after photobleaching. And the way this works is, and this is used commonly in the gap junction field, is using fluorescence microscopy. So we're looking down a microscope at fluorescently labeled cells. And if you imagine the cell on the left-hand side of the slide here at the top, imagine it has a green fluorescent protein that's mobile, it's able to move around the cell. If we draw an area on our microscope, we define an area that we'd like to look at in a bit more closely, we can fire a high-power laser at that area, and it causes the fluorescence to bleach. So lots of fluorescent compounds, they will become less fluorescent when you give them, when you shine a very bright light on them. And that's what happens in this box. It becomes very pale. But because the protein is able to move, it floats back into that area. And so if you look inside that box, gradually the green recovers. So that's fluorescence recovery after photobleaching. That's what it stands for. And this tells you a lot about what the molecules can do. Can they move? Are they free to move around? So we can apply this sort of technique between cells and to see if they're talking directly to each other. And the way we can do this is using different size fluorescent dyes. So imagine you have two cells and they are connected by a gap junction, and you bleach the one on the right-hand side. Dyes that are small enough to get through the gap junction will be able to get back through again, and the fluorescence will recover. But if the cells aren't joined by gap junctions, they won't recover. So we've applied this technique to look at blood clots. And what you're looking at here is fluorescently labeled platelets. So they have a fluorescent dye inside them. And they've stuck to each other to form a thrombus. We form this down the microscope using microfluidic flow cells. And what we do is to cause an area of bleaching, and that causes the fluorescence to disappear over here on the graph. And then very quickly, we're able to see fluorescence recovers. So this fluorescence is coming from the cells around. But if we do this in the presence of our inhibitor of gap junctions, it prevents this from happening. So this shows that platelets are talking to each other directly through gap junctions. But why might this be important? I mentioned earlier that heart cells coordinate their activity through gap junction type communication. And that is really what gap junctions are for. They're there to coordinate the responses between cells so that cells that are sitting next to each other can do similar things at similar times. And this is really important in lots of different tissues. So what might platelets do that requires coordination? Well, there's one thing that we thought of, and that's clot retraction. So when a blood clot forms, if we were to take a blood sample from my arm and put it in a tube and just put it there and leave it for 10 minutes, within about 10 minutes, it will become completely solid. So it becomes coagulated, a completely solid blood. If we leave it for another hour, the clot will get smaller, it will contract, and we think that this is to help wound repair if it happens inside the body. But this process, we know, is controlled by the platelets that are trapped inside the clot. So we asked what happens to clot retraction in the presence of our inhibitor. And so this is how we study it. We literally form blood clots inside tubes. We incubate them over time in the presence of the inhibitors. And then at the end of that time, we look at how big the clots are. And in this case, we've done it just by weighing the clots at the end. And so a heavier clot has got more liquid left in it, so it's less retracted. So what this experiment shows us is that clot retraction is inhibited if we inhibit connexins. So we've done a lot of work to try and work out how these work, and I'm not going to go through that in detail now, but we now understand that connexins can control cell communication mechanisms within either cell on either side. And we've looked at the ability to regulate kinases and other enzymes that are really important in determining and making platelets function as they do. But we've been particularly interested in whether we can visualize this happening in real time. And so to do this, we've adapted a technique we would traditionally use by flow cytometry, as I showed you earlier on, where I showed you an experiment where we loaded platelets with fluorescent dyes, and then we stimulated them, and we looked to see whether the dye was released. Well, we can take that one step further and use this in fluorescence video microscopy. So in this case, what we've done is to take some platelets from the blood, and we've loaded them with a fluorescent dye, so the dye is trapped inside them. But we've also labelled the outside of these cells with an antibody that tells us that they are platelets. And we want to do that because we're going to put them back into the blood where there's lots of other cells as well. Most of the platelets in the blood are not labelled, but we've labelled about 10% of them and we put them back. We then form thrombi using microfluidic flow cells under flow conditions, a bit like the conditions that would be experienced by platelets in the circulation. And what we're then able to do is to look and see where the fluorescence goes. And if we find the fluorescent dye in a cell, in platelets that don't have the antibodies on them, that means the dye must have come from another platelet that did have an antibody on the outside of it. Maybe it will be clearer if I show you this in what actually we see down the microscope. So in red, we see the donor cells, and in green, we can see the cells that have now got the fluorescent dye inside them. So this is looking down on platelets forming thrombi in microfluidic flow cells using high-speed microscopy. And each of the green cells you can see here has obtained its green dye from other platelets. So this demonstrates that it's really happening within thrombi. And we're able to actually then look and see what happens when we use inhibitors of connexin. So CBX is carbonoxylone, which is one of the general inhibitors I mentioned earlier on. And you can see here that the number of green cells goes down. You can see it just by looking at the slide. We can see this sort of shown graphically here. But we can do this also with our various GAP peptides, and we can see that it reduces the signal. And if we add all of our peptides together to the 3 connexins that we know are on platelets and are important, we can see a very similar level of inhibition to what we see when we use the general inhibitor here. So that tells us that these 3 connexins are really important in regulating platelet function. One other thing that might be relevant as well to situations where blood clots are forming inside vessels is that causes changes in flow. And it can cause what we call high shear conditions. So there's a lot of stress exerted on the cells as they stick to the surface. And what we find is that if we flow blood faster, which effectively increases the stress on them, we get more transfer of dye between the cells. So this also looks like it's a process that's affected by the level of flow, which is a strong indicator that this might contribute to unwanted platelet activation where you've got vessels that are becoming occluded through lesions in them. So I wanted to finish with one new story that's associated with this, just a couple of slides, because we're interested in understanding how these particular channels open and close. Because if we can understand how they open and close, or how they're moved around the cell to make them able to function or not function, then we will have new ways to turn down their behavior, turn down their function on cells, and potentially use this as a way of preventing thrombosis and preventing heart attacks and strokes. So this particular piece of work started by a really odd observation we had. This is an immunoblot, so this is using antibodies to detect connexin, in this case connexin 62, from human platelets. And the left-hand side here is platelets that are resting. They've not seen anything that would activate them. But these 2 columns next to the 2 lanes next to this are platelets, the connexin 62 in platelets following their activation. So we've activated them with thrombin or with collagen. That's not really important, but they're activated platelets. And what you notice is that the level of protein goes down, which is a bit odd, but we didn't quite know what was going on here. And eventually we worked out that the protein is being cleaved, it's being chopped. And we did this using antibodies and various techniques, but what we discovered was that this cleavage of the protein, which resulted in a smaller fragment appearing at the bottom of our gels that we ran, was dependent on calcium. So if we had high levels of calcium, the cleavage happened, but we could inhibit it if we got rid of the calcium. Now there are a range of enzymes, usually inside cells, that are calcium sensitive and able to chop up proteins. And some of these proteins are really important for controlling platelet function. They're involved in a lot of the communication machinery inside platelets. So we thought about one of these, whether this might be an explanation. And so we did the same experiments in the presence of an inhibitor called calpeptin. And calpeptin inhibits an enzyme called calpain. And what hopefully you can see here is that the addition of calpeptin prevents the protein from being cleaved. So this tells us that the enzyme that's causing the cleavage, and it is cleavage, is calpain. So we asked the question, are there sequences on the connexins that could be cut by calpain? And to do this, we used various online tools, and we were able to predict a calpain cleavage site on the protein. And this is actually a docked model of calpain here in blue or turquoise, docked onto connexin 62. So we can see that it should be able to interact. But the really confusing thing is that the cleavage site is on the outside of the cell, which isn't where you'd expect the calpain to be able to cut it, because the calpain is normally found on the inside of cells. And what we were able to show is that platelets release calpain. When platelets are activated, they release this enzyme. We're not quite sure how at this point, but they release this enzyme. And if we inhibit it, we can prevent, we can basically reduce gap junction function. So this tells us that the cleavage is important for regulating how the channels work. And so what we did to explore this was to make a peptide that is the same sequence as the cleavage site on the connexin. We call this a decoy peptide because it should bind to calpain, and we've shown that it does, and it should prevent the calpain from cleaving the connexin. And so that's this peptide here. We call it 62-PEPT-NT. And on the left-hand side, this is actually really important. If you use peptides as tools like this, you need a control. And the control is the same amino acids, but in a different sequence that we've checked won't bind to anything else. And basically what we see in the presence of our decoy peptide is a decrease in the transfer of dye between platelets in thrombi. So this tells us that calpain cleavage opens the channel. That's what we would predict. So we've done some molecular modelling using AlphaFold 2, because everyone can use it now. It's available for everyone to play with. And this is what we saw. Actually, to be fair, we do have a structural biologist on the team who knows what they're doing with these, because they're dangerous tools if you don't know how to use them. So basically, on the left-hand side, you can see non-cleaved hemichannel or connexin ring. And on the right-hand side, you can see it cleaved. And hopefully you can see the difference between the two is the channel opens up. So we think this is a new mechanism that regulates connexins and might be something that we could target to reduce platelet function. So just to summarize, hopefully I've convinced you that platelets communicate, but not only do they receive signals from each other from chemicals that they release, or they receive signals from tissue damage or everything that's going on around them, they also are able to communicate directly and pass signals from the inside of one platelet to the next. This is a phenomenon that is completely new in the area of blood cells. It's not something that is well recognized. It's well recognized in tissues, so this opens up some interesting possibilities for new ways that we could potentially develop drugs to treat disease. I just need to acknowledge lots of people who've been involved in this, and as Professor Salazar said, that one of the key things with running any lab is the pleasure you get from seeing the number of people working together, training, traveling the world, going and seeing other people and working together. So I'm not going to go through them all in detail, but there's a lot of people on that slide who've contributed to this story, both at Reading, colleagues and also members of my lab, and also colleagues around the world who've been very generous with their time and reagents. And of course, similar situation to in Chile, we have to pay for the research as well, and so these are the funders of the research that we do. I should stop there. I hope I haven't gone on too long, and stop there and be very happy to take any questions or chat afterwards. Thank you.